Picture this! You're a 46-year-old woman from Dorset, England. You've been told your stage 4 melanoma is terminal. The doctors have said there's not much more that they can do. You have weeks, maybe months. You're making your peace with mortality. You're saying your goodbyes. You're putting your affairs in order. And then, 6 weeks after a single treatment, your tumors completely disappear. Every single one of them. That's not science fiction. That's not a miracle. That's Zoe Phillips' real story from 2023. And it's happening because of a treatment that's been 35 years in the making. February 16th, 2024, the FDA finally approved Lifileucel. A mouthful of a name for a revolutionary treatment that uses your own immune cells as a living medicine against cancer. It's the first time a cellular therapy has ever been approved for solid tumors, and it represents one of the most significant milestones in oncology in the past decade. But here's what makes this story even more remarkable. The journey started back in 1986, when a surgeon named Steven Rosenberg at the National Cancer Institute had a wild idea. What if we could take the immune cells that are already inside tumors, the ones that try to fight cancer, but then get exhausted, harvest them, wake them up, grow billions of copies, and send them back into battle? It sounded almost too simple, and yet, here we are, nearly 4 decades later, watching that simple idea transform into a commercial reality that's giving hope to patients who've run out of options. Welcome to Apprentice.io's Industry Insights. I'm Frank Maggiore, and today we're diving deep into the world of tumor infiltrating lymphocyte therapy, TIL therapy, for those of us who prefer not to twist our tongues, and exploring how this groundbreaking treatment is reshaping cancer care, pharmaceutical manufacturing, and the very definition of personalized medicines. Now, I know what some of you are thinking. Another cancer immunotherapy? Haven't we heard this story before with checkpoint inhibitors and CAR T cells? And yes, we have. But TIL therapy is different in some fundamental ways that make it uniquely suited to tackle cancers that other treatments can't touch. This isn't just about the science, though we'll get into the fascinating biology of how your immune cells can be trained to become precision-guided missiles against cancer. It's also about the manufacturing challenge of creating billions of cells for each individual patient. The role of cloud-based manufacturing execution systems in ensuring the right therapy gets to the right person. How artificial intelligence is supercharging these treatments, and ultimately, what this means for the future of medicine itself. So whether you're a pharma manufacturing professional wondering how on earth you scale personalized cell therapy, a healthcare worker curious about the latest treatment options, or someone just fascinated by the intersection of biology, technology, and hope, you're in the right place. Let's talk about the living army within. Alright, let's start with the basics, because TIL therapy is one of those topics where understanding the foundation makes everything else click into place. Your immune system is, put it simply, amazing. Every day, it's patrolling your body looking for threats, bacteria, viruses, and yes, cancer cells. And here's something that surprises a lot of people. Your immune system is actually finding and destroying cancer cells all the time. We all have cells that go rogue occasionally, and our immune system quietly takes care of them. It's the ultimate homeland security operation happening 24/7. But sometimes, cancer gets smart. It evolves ways to hide from your immune system. Or worse, it creates an environment, what we call the tumor microenvironment, that basically tells your immune cells to stand down. It's like cancer sends out a signal saying, nothing to see here, move along. These are not the droids you're looking for. And your immune cells, even though they've recognized the threat and infiltrated the tumor, get exhausted and switch off. These immune cells that make it into the tumor are called tumor infiltrating lymphocytes, or TILs. They're primary T cells, the special forces of your immune system, and they've actually done the hardest part already. They found the cancer and recognized it as foreign. They're literally inside the tumor trying to kill it. They're just tired, deactivated, outnumbered. TIL therapy is essentially an extraction, reinforcement, and redeployment operation. Here's the process. First, surgeons remove a piece of the tumor, about the size of a grape. Inside that tumor are millions of these exhausted TILs. Scientists isolate those cells, put them in a lab, and give them everything they need to wake up and multiply. Growth factors, nutrients, the works. Over the course of a few weeks, those millions of cells become billions. We're talking about 20 to 50 billion cells from a single sample. Then comes the crucial part. Before those cells are infused back into the patient receives chemotherapy to temporarily suppress their existing immune system. That might sound counterintuitive. Why suppress the immune system when we're trying to boost it? But it's like clearing the battlefield before sending in fresh troops. It removes suppressor cells and makes room for the TILs to expand and function. The billions of supercharged TILs are infused back into the patient's bloodstream. These cells remember the cancer, they recognize it, and now they have the numbers and energy to actually do something about it. They circulate through the body, find tumor cells wherever they're hiding, and then they attack. Now, you might be thinking, this sounds a lot like CAR T-cell therapy, which you probably heard about for blood cancers like leukemia and lymphoma. Both are cellular therapies, both use T-cells, and both are personalized to each patient. But there are some critical differences that make TILs particularly well suited for solid tumors like melanoma, lung cancer, and breast cancer. CAR T cells come from the patient's blood. They're collected, genetically engineered in the lab to express chimeric antigen receptor. That's the CAR and CAR T. That targets a specific protein on cancer cells. It's like giving the T cells GPS coordinates to a single target. This works great for blood cancers where that target protein is consistent and accessible. But solid tumors, they're heterogeneous. Different cells in the same tumor can have different targets. They also have physical barriers and immunosuppressive environments that make them harder to penetrate. TILs, on the other hand, come from inside the tumor itself. They're already there, already naturally selected to recognize the cancer's antigens, including neoantigens, which are unique mutations specific to that patient's tumor. TILs are polyclonal, meaning they target multiple different antigens simultaneously. It's not GPS to one location, it's a heat-seeking missile array that can find cancer cells even if they try to hide by changing their appearance. And here's the really elegant part. TILs don't require genetic engineering to work. They're naturally equipped to recognize the cancer. Now, scientists are absolutely working on genetically modified TILs. We'll get to that later. But the basic TIL product is your own cells, just amplified. So where are we using TIL therapy today? Melanoma is the leading indication. That's where the most clinical data exists, and that's where Lifileucel got its FDA approval. But the clinical trial landscape is exploding. We're seeing promising results in cervical cancer, particularly HPV-driven cases. Non-small cell lung cancer, especially in patients whose diseases progress after checkpoint inhibitors. Breast cancer, with some remarkable complete responses in individual patients. Ovarian cancer, head and neck cancers, gastrointestinal cancers, including some colorectal cancers with specific mutations. There are over 100 clinical trials currently underway globally, testing TILs across different cancer types, in different combinations, and at different stages of disease. The patient journey typically takes about 10 weeks from start to finish. It begins with that tumor biopsy, which can be straightforward or complex depending on where the tumor is located. The sample is shipped overnight to a specialized manufacturing facility where the TILs are isolated and expanded over about 3 to 5 weeks. During this time, the patient isn't hospitalized. They're living their life. When the TILs are ready, the patient is admitted for lymphodepleting chemotherapy, usually cyclophosphamide and fludarabine for about a week. Then comes the TIL infusion itself, which takes about 2 hours. After that, patients receive a series of doses of medication called interleukin-2 to activate and support the TILs as they expand in the body. The hospital stay is usually just a few days for monitoring, and then several weeks of recovery as the immune system rebuilds. One treatment, one time, and for patients who respond, those TILs can stay active for years, continuing to patrol and protect. It's a demanding process, absolutely. It requires patients to be healthy enough to tolerate high-dose chemotherapy, and it requires sophisticated infrastructure. But for patients who have exhausted other options, or for those whose cancers are particularly aggressive, it's opening a door that was previously closed. And that brings us to the really exciting part, the technology that makes this all possible. Let's pull back the curtain on how TIL therapy actually gets made, because this is where biology meets engineering in some truly fascinating ways. And I'll warn you up front, this gets complex, but stick with me, because understanding the manufacturing process is crucial to understanding why cloud-based systems and digital transformation are so critical to this field. The TIL manufacturing process is essentially a highly controlled biological production line, and every step has to be executed with precision. Let's walk through it. Step 1. Tumor resection and processing. A surgeon removes tumor tissue from the patient, either through a biopsy or during a larger surgical procedure. This tissue needs to stay viable, so it's immediately preserved and chilled. Time is critical here. These cells need to stay alive. The sample is typically shipped overnight to a centralized manufacturing facility. And right here, we encounter our first major manufacturing challenge: chain of identity. You absolutely cannot mix up patient samples, ever, full stop. The consequences would be catastrophic. Step 2. Once the tumor arrives at the manufacturing facility, scientists literally dissect it into tiny fragments. We're talking pieces just a few millimeters across. TILs are then isolated from these tumor fragments. Sometimes this involves enzymatic digestion to break down the tumor tissue and release the cells. Other times, the fragments themselves are cultured directly, allowing TILs to migrate out. Different manufacturers have different protocols, but the goal is the same: get those TILs out and into culture. Step 3, the pre-rep or initial expansion phase. The isolated TILs are cultured in a specialized growth media containing interleukin-2 or IL-2, which is a cytokine that promotes T-cell growth and proliferation. This initial culture can take 1 to 2 weeks, during which the TILs start to expand in number. Scientists are monitoring these cultures constantly, checking for viability, growth rates, and signs of contamination. Step 4, Rapid Expansion Protocol, or REP. This is where the real magic happens. The TILs undergo a rapid expansion process using anti-CD3 antibodies and high doses of interleukin-2, often with irradiated feeder cells that act like support systems. Over the course of about 2 weeks, the cells numbers explode. We're going from millions to tens of billions of cells. Modern protocols like Iovance's Gen 2 process have streamlined this to about 22 days total, down from several weeks in older protocols. Now, here's where the manufacturing innovation is really accelerating things. Companies are moving from open systems, where cells are cultured in flasks and plates that require manual handling in clean rooms, to closed, automated bioreactor systems. These systems reduce contamination risk, decrease hands-on labor, and enable more consistent production. Step 5, quality control and testing. Before those billions of cells can be released for patient infusion, they undergo rigorous testing. Sterility testing to ensure no bacterial or fungal contamination. Endotoxin testing. Viability testing. What percentage of the cells are alive and functional? Potency testing. Can these cells actually kill tumor cells in a lab dish? Immunophenotyping, what types of T cells are present, and identity testing, confirmation that these cells are definitely from the correct patient. This is not a quick process. Some of these tests take days to complete, and if anything fails, that entire batch is rejected. Months of work, all gone. Step 6, cryopreservation and shipping. Once the TILs pass all quality checks, they're formulated in a final infusion solution, typically with a cryoprotectant, and then frozen. They're then shipped back to the treating hospital in specialized containers that maintain ultra-low temperatures. Chain of custody documentation follows every step. Now, while all this is happening at the manufacturing facility, we haven't forgotten about the patient. The clinical team is preparing the patient for treatment, coordinating the timing of lymphodepleting chemotherapy so that it aligns when the TIL product will be ready for infusion. Let me emphasize something here. This is autologous therapy. Every single patient gets their own unique product made from their own cells. This isn't like manufacturing a drug where you make millions of identical doses. This is mass customization at the cellular level. It's more like custom manufacturing 747s than it is like bottling aspirin. And this is where the next generation of TIL therapy gets really interesting. Scientists are now engineering TILs to make them even more effective. Let me give you a few examples. CRISPR gene editing. Companies like KSQ Therapeutics are using CRISPR to knock out specific genes in TILs that inhibit their function. Their lead product, KSQ-004EX, inactivates 2 genes, SOCS1 and REGNASE1. These genes normally act as brakes on T-cell activation. Remove the brakes and you get TILs that are more aggressive, more persistent, and more effective at killing tumors. The first patient was dosed with this CRISPR-engineered TIL in April 2025, and the field is watching closely. Membrane-bound cytokines, one of the major drawbacks of current TIL therapy, is the need for the high doses of interleukin-2 after infusion, which can cause significant side effects: fever, low blood pressure, fluid retention. Obsidian Therapeutics has developed OBX-115, also called Cytotil-15, which engineers TILs to express membrane-bound interleukin-15. This means the TILs essentially bring their own growth and activation signal with them. Early data suggests this eliminates the need for systemic interleukin-2 while maintaining or even improving efficacy. That's a game changer for patient tolerability. But here's the catch. Every additional manipulation step adds complexity, time, and potential failure points to the manufacturing process. It also adds regulatory scrutiny. These engineered products will need to demonstrate that the benefits outweigh the additional risks and costs. The technology behind TIL therapy is a beautiful fusion of immunology, cell biology, bioprocess engineering, and information technology. And as we'll see next, this convergence is absolutely game-changing for patients. Now we get to the heart of why TIL therapy is generating so much excitement. And it's not just the cool science, it's the clinical results. It's what's happening to real patients. Let's start with some context. Melanoma treatment has come a long way in the past 15 years. Before 2011, if you had advanced metastatic melanoma, your median survival was about 6 to 9 months. It was a death sentence. Then came the revolution, BRAF inhibitors for patients with BRAF mutations, and checkpoint inhibitors like ipilimumab, nivolumab, and pembrolizumab. These were game changers. Suddenly we were seeing response rates of 40 to 60% with combination immunotherapies. Some patients were achieving durable, complete responses, being cancer-free for years. This was unprecedented. But, and this is a big but, that means 40 to 60% of patients aren't responding to frontline therapy. And of those who do respond, about a third will eventually progress. So, we still have a massive unmet need for patients whose disease doesn't respond to checkpoint inhibitors or who progress after initial response. Enter TIL therapy. The pivotal clinical trial that led to the FDA approval was the C11401 study, which enrolled patients with advanced melanoma who had already progressed on checkpoint inhibitors and, if they had BRAF mutations, on BRAF inhibitors too. These were heavily pretreated patients who had essentially run out of standard options. Out of 73 patients treated in the approved dose cohort, the overall response rate was 31.5%. Now you might think, only 31%, that doesn't sound that impressive. But context matters. These patients had failed multiple prior therapies. Many oncologists would have expected response rates in the single digits with whatever remaining options existed. But here's where it gets really interesting: the durability of responses. Among patients who responded, almost half maintained their response for more than 24 months. Some patients saw their tumors continue to shrink over time, going from partial responses to complete responses, even a year after treatment. And remember, this was a one-time treatment. They weren't on continuous therapy. Fast forward to the 5-year follow-up data presented at the 2025 ASCO annual meeting just this past June. Tumor shrinkage was observed in nearly 80% of patients. Almost 1/3 of responders were still in response at 5 years. And critically, 19.7% of patients were alive at 5 years. Think about that. Nearly 1 in 5 patients with heavily pretreated advanced melanoma alive 5 years after a single treatment. Without TIL therapy, most of these patients would have had life expectancy measured in months. And the side effect profile? Most of the toxicity came from the lymphodepleting chemotherapy or the interleukin-2, both of which were manageable and temporary. No new or unexpected long-term toxicities emerged over 5 years of follow-up. But wait, it gets better. The M14 TIL trial in Europe randomized patients to either TIL therapy or ipilimumab. The results, published in the New England Journal of Medicine, showed that progression-free survival was more than double with TIL therapy compared to ipilimumab. The separation of survival curves happened within 6 months and continued to diverge over time. Patients in the TIL arm lived a median of 25.8 months compared to 18.9 months with ipilimumab. These are phase 3 randomized data showing superiority. That's the gold standard of clinical evidence. Now, let's talk about expanding beyond melanoma, because this is where the future gets really exciting. Non-small cell lung cancer. About 20 to 25% of patients with non-small cell lung cancer respond to checkpoint inhibitors. For the rest, options are limited. Early phase trials of TIL therapy in checkpoint inhibitor-resistant lung cancer have shown response rates in the 20 to 30% range. One patient in a phase 1 trial at Moffitt Cancer Center had complete regression of disease that lasted for years. Clinical trials combining TILs with pembrolizumab are now underway. For cervical cancer, HPV-driven cancers are particularly interesting for TIL therapy because HPV proteins represent non-self antigens that TILs can recognize. Clinical trials have shown encouraging response rates in patients with recurrent metastatic cervical carcinoma who had progressed on standard therapies. For breast cancer, this is fascinating. Breast cancer has historically been considered cold, meaning not very immunogenic, not a lot of immune infiltration, but there have been individual patients in TIL trials who had complete responses. As researchers learn to better identify which breast cancer patients have tumors with higher immune infiltration and mutational burden, TIL therapy could open up a whole new treatment avenue. For ovarian cancer, head and neck cancers, and gastrointestinal cancers, all of these are currently being explored in clinical trials with early signals of activity. And then there are the enhancement strategies. Remember those engineered TILs we talked about? The early clinical data on OBX-115, the interleukin-15 equipped TIL, suggests it's achieving response rates comparable to or better than standard TILs, but without the need for toxic interleukin-2. If that holds up in larger trials, it dramatically improves the patient experience and expands the eligible patient population to include those who couldn't tolerate interleukin-2. One of the most compelling aspects of TIL therapy is the patient selection evolution. Initially, TIL therapy was positioned as a last resort, something you try when everything else has failed. But as we get better at predicting who will respond, using biomarkers like tumor mutational burden, PD-L1 expression, and immune infiltration patterns, there's growing interest in moving TIL therapy earlier in the treatment paradigm. The TILVANCE 301 trial, which is currently enrolling, is testing TILs plus pembrolizumab as frontline treatment for advanced melanomas, comparing it to combination checkpoint inhibitor therapy. If TILs show comparable or superior efficacy with a better safety profile, we could see a fundamental shift in how advanced melanoma is treated. There's another aspect of TIL therapy that's worth highlighting: the psychology of one-time treatment. Many cancer therapies require continuous administration. You're on treatment until progression or intolerable side effects. That means monthly infusions, daily pills, constant medical appointments, perpetual reminders that you're a cancer patient. With TIL therapy, you go through an intense 10-week process, and then you're done. You're monitored, obviously, but you're not actively being treated. For patients who respond, there's something psychologically powerful about knowing your own cells are out there doing the work without you needing to do anything. Is TIL therapy a panacea? No, not every patient responds, and we're still learning who the best candidates are. But for a significant subset of patients with solid tumors, particularly those who've exhausted standard options, TIL therapy is providing durable responses and extending lives in ways that were simply not possible just a decade ago. And here's the thing, we're still early. Lifileucel is the 1.0 version of TIL therapy. The 2.0, 3.0, and 4.0 versions are in the pipeline. As we learn more, engineer smarter, and refine the process, the efficacy will only continue to improve. But none of this can scale without the right manufacturing infrastructure, and that's where things get really interesting from an industry perspective. Alright, let's talk about the elephant in the clean room. Manufacturing Execution Systems, or MES. I know, manufacturing software doesn't sound as exciting as cancer-fighting supercells, but trust me, if you're in pharmaceutical manufacturing, biotech, or cell and gene therapy, this is where the rubber really meets the road. And frankly, if you don't get the manufacturing right, it doesn't matter how brilliant the science is. The product won't reach its patients. So, let's set the stage with the challenge. TIL therapy is autologous, which means each batch is specifically made for one patient from their own cells. You can't manufacture 10,000 doses and put them on a shelf. You can't even manufacture 10 doses for 10 different patients in the same bioreactor at the same time. Every single patient gets their own manufacturing run. Now, imagine your Iovance Biotherapeutics or any TIL manufacturer trying to scale to commercial production. You've got an FDA approval. You've got patients enrolling. Oncologists are prescribing your therapy. You need to go from manufacturing dozens of batches per year in clinical trials to hundreds or even potentially thousands of batches per year at commercial scale. Each batch takes 3 to 5 weeks to manufacture. Each batch has dozens of process steps, hundreds of documentation points, multiple quality control checkpoints, and an absolute zero tolerance policy for mix-up. So how do you manage that? With paper batch records. Short answer, you don't. You can't. Paper-based systems that work fine when you're doing 20 batches a year in a clinical trial collapse under the weight of commercial-scale autologous manufacturing. This is where modern cloud-based manufacturing execution systems come in. Platforms like Apprentice.io's Tempo MES that are purpose-built for cell and gene therapy manufacturing. Let's talk about the most critical requirement, chain of identity, or COI. From the moment that the tumor biopsy arrives at your facility until the final TIL product is infused back into the patient, you must maintain absolute certainty about whose cells you're working with. A traditional paper-based system might use handwritten labels, manual logbooks, and operator signatures. It works until it doesn't. A nurse reads patient 1023 as patient 1028. A label falls off a culture flask. A batch record gets filed in the wrong folder. In autologous cell therapy, there is no margin for error. Giving the wrong cells to the wrong patient could be fatal. Not only does that patient not get their therapy, but they're receiving foreign cells that their immune systems will reject, potentially causing severe complications. Cloud-based manufacturing execution systems address this through multiple layers of protection. Electronic chain of custody. Every movement of material is logged electronically with timestamps and user IDs. When a tumor sample is received, it's scanned into the system. When it's moved to processing, it's scanned. When cells are transferred to a bioreactor, they're scanned. At every handoff, there's barcode verification or RFID tracking. The system has built-in guardrails. It won't let you proceed to the next step if identity verification fails. Try to add media to bioreactor A when you should be working on bioreactor B, the system blocks you and alerts a supervisor. This isn't about being overly cautious. This is about being appropriately cautious. Real-time monitoring of process parameters are captured automatically from equipment. Temperature, pH, dissolved oxygen, cell counts, all logged in real time with no opportunity for transcription errors or retroactive data filing. With electronic batch records, the MES generates complete, tamper-proof batch records automatically. Every action, every deviation, every check is documented as it happens. No more staying late after a run to fill out the paperwork. No more, we'll document it later. No more illegible handwriting. Now, let's talk about the operator experience, because this is where cloud-based systems really shine compared to older legacy MES implementations. Traditional MES systems were often paper on glass, basically taking a paper procedure and putting it on a static PDF on a screen. Not much better than actual paper, and in some ways, worse because you lose the flexibility of paper. Modern cloud-based MES platforms like Apprentice.io's Tempo take a completely different approach. It's a mobile-first design. Operators can use tablets, smartphones, or even smart glasses to access procedures wherever they're working. No more running back and forth between a fixed computer terminal and the biosafety cabinet or a bioreactor. The instructions, they come with you. With augmented reality capabilities, some systems now support AR headsets that overlay instructions directly onto the operator's field of view. Imagine you're a technician working in a clean room with dozens of identical cell culture flasks. With AR, you can see digital labels and instructions floating next to each flask, eliminating any confusion. With voice-enabled operation, you can work hands-free when you're gloved and busy with sterile procedures. Voice commands can be a game changer. Tempo, record a temperature reading, 37.2 degrees Celsius. The system logs it. No touching a keyboard with potentially contaminated gloves. By leveraging multimedia instructions, instead of just text procedures, you can include photos, videos, or even 3D models. An operator can watch a video of exactly how to disassociate a tumor sample or load a bioreactor, then perform the procedure themselves with confidence. Now let's talk about equipment integration and automation, because manual data entry is not only tedious, it's error-prone. Modern manufacturing execution systems integrate directly with manufacturing equipment through industry-standard protocols. When a bioreactor reaches target cell density, that data flows automatically into the MES. When an incubator temperature alarm goes off, it's logged. When a fill-finish machine dispenses the final product into vials, the volumes and lot numbers are captured. Another huge advantage of cloud-based systems is scalability and flexibility. As your production volume increases, you don't need to install new servers or upgrade local infrastructure. The cloud scales with you. Need to spin up a second manufacturing site on another continent? Your MES is already there. You just configure it for the new facility. And here's something that's becoming increasingly important: data analytics and continuous improvement. Traditional manufacturing captures data, but that data sits in paper records or in isolated databases. Cloud-based manufacturing execution systems centralize that data in formats that enable sophisticated analysis. You can now identify trends, correlate process parameters with product quality, benchmark operators and shifts, and continuously optimize your manufacturing process. Let me give you a concrete example. Suppose you notice that batches processed on Monday mornings have slightly lower cell viability than batches processed later in the week. With paper records, you might never notice this pattern. With a cloud-based manufacturing execution system that's aggregating and analyzing that data, this trend becomes visible. You investigate and discover that your cleaning crew is using a different disinfectant on Sunday nights that's leaving residue. Problem identified, problem solved. Product quality improves. Now, I'd be remiss if I didn't address the concern that always comes up with cloud-based systems in pharma: regulatory compliance and data security. Good news, cloud systems, when implemented correctly, are often more compliant and more secure than on-prem systems. From a compliance standpoint, cloud-based manufacturing execution systems are designed to meet 21 CFR Part 11 requirements. GMP standards, they have built-in audit trails, electronic signatures, data integrity controls, and security measures that would be expensive and complex to implement in a homegrown system. From a security standpoint, reputable cloud providers are investing millions of dollars into security infrastructure, encryption, intrusion detection, redundancy, disaster recovery, far more than most individual companies could afford to implement themselves. Just ask the Korean government when their on-prem data center caught fire. Let me bring this back to TIL therapy specifically. Companies like Apprentice.io have built MES platforms with cell and gene therapy manufacturing in mind. They understand the unique challenges: managing autologous batches, maintaining chain of identity, integrating with specialized equipment like bioreactors and cell counters, handling cryopreservation and thaw processes, managing shipping logistics. These aren't generic manufacturing systems adapted for biopharma. They're purpose-built solutions that speak the language of cell therapy. And here's the thing: as TIL therapy continues to expand, as we go from one approved product to 5, from 5 to 20, and companies that have robust, scalable, validated manufacturing infrastructure will be the ones that succeed. Because ultimately, the best science in the world doesn't help patients if you can't manufacture the product reliably, at scale, and with absolute quality assurance. Cloud-based manufacturing execution systems aren't just nice-to-have tools for modern pharmaceutical manufacturing. For autologous cell therapy, they're essential. They're the difference between pilot-scale production and commercial success. They're the difference between helping dozens of patients and helping thousands. Alright, buckle up, because this is where we get into the really exciting developments happening right now in TIL therapies. We're not talking about future possibilities, we're talking about clinical trials enrolling today, approvals happening this year, and technologies that are actively transforming the field. Let's start with the landmark moment, February 16th, 2024. The FDA grants accelerated approval to Lifileucel, marketed as Amtagvi by Iovance Biotherapeutics. This is the first TIL therapy approved anywhere in the world. It's the first cellular therapy approved for any solid tumor. It's a genuine historic milestone. The approval covers adult patients with unresectable or metastatic melanoma who have been previously treated with a PD-1 blocking antibody, and if they're BRAF V600 positive, a BRAF inhibitor. Essentially, patients who progress on standard therapies. Now, here's the thing about accelerated approval. It's based on single-arm trials showing objective response rate and duration of response. The FDA has required a confirmatory trial to verify clinical benefit, which is currently ongoing. But the point is, the therapy is available now. Patients can receive it today at authorized treatment centers across the United States. But Amtagvi is just the beginning. The TIL therapy pipeline is absolutely exploding. According to market research, there are more than 75 TIL-based therapies in development. and over 100 clinical trials underway globally. The market is projected to grow from $87 million in 2024 to an astounding $5 billion by 2035. That's a compound annual growth rate of 40%. Let me highlight some of the most exciting programs currently in development. Obsidian Therapeutics with their OBX-115. This is the membrane-bound interleukin-15 engineered TIL I mentioned earlier. In June 2025, Obsidian announced initial phase 1 safety and efficacy data from the AGNI-01 study. The early results suggest that the Cyto-TIL-15 is safe and showing clinical activity without requiring systemic interleukin-2 administration. If confirmed in larger trials, this would be a massive improvement in patient experience. No more interleukin-2 toxicity, no fever, no hypertension, no capillary leak syndrome. This means more patients would be eligible for treatment, and those who receive it would have an easier recovery. KSQ Therapeutics, with their KSQ-004EX in April 2025, marked the dosing of the first patient with this CRISPR-engineered TIL. KSQ-004EX has the SOCS1 and Regnase-1 genes inactivated using CRISPR-Cas9. Preclinical data suggests these modifications result in TILs with enhanced persistence and function. The field is watching closely because this represents the next generation of TIL therapies, combining natural tumor recognition with precision gene editing to enhance performance. Academic centers are also helping to lead the charge. The National Cancer Institute, Memorial Sloan Kettering Cancer Center, the Royal Marsden NHS Foundation Trust, the Netherlands Cancer Institute, Copenhagen University Hospital. These institutions continue to lead cutting-edge TIL research. They're testing TILs in new cancer types, in new combinations, and with enhancement strategies. Now let's talk about the combination strategies, because this is where TIL therapy gets really interesting. They're specifically looking into TIL with checkpoint inhibitors. There's a strong rationale for combining TIL with PD-1 or PD-L1 blocking antibodies. The checkpoint inhibitor can help prevent TILs from becoming exhausted after infusion, potentially improving their durability. Several trials are testing Lifileucel plus pembrolizumab. The IOV-COM-202 trial is evaluating this combination in treatment-naive melanoma patients, potentially moving TIL therapy to the front line. Even more creatively, TILs are being combined with oncolytic viruses. Oncolytic viruses, like TILT-123, selectively infect and kill cancer cells, causing them to release antigens and inflammatory signals. This can heat up cold tumors, making them more visible to TILs. A trial combining TILT-123 with TILs in checkpoint inhibitor refractory melanoma is currently underway. Now let's talk about artificial intelligence and how it's supercharging TIL therapies. This is one of the most exciting developments, and it's happening at multiple levels. When it comes to patient selection and predictive modeling, not every patient responds to TIL therapy, and we love to know in advance who the best candidates are. AI models are being trained on tumor biopsies, imaging data, and clinical characteristics to predict who's most likely to respond. For example, algorithms can analyze digital pathology slides to quantify the degree and type of immune infiltration, assess tumor mutational burden, and identify biomarkers associated with response. This allows clinicians to have informed discussions with patients about expected benefits before committing to the treatment. In manufacturing optimization, AI is being used to monitor and optimize the cell expansion process in real time. Machine learning algorithms can predict when cells have reached optimal growth, when they need media changes, and when they're ready to harvest. This reduces batch failures and improves product consistency. The real cutting-edge stuff centers around enhanced TIL design. Researchers at Technical University of Denmark and Scripps Research Institute published a study in July 2025 describing an AI platform that designs mini binders, small engineered proteins that can be added to TILs to help them recognize and bind to cancer cells more effectively. The AI identified and designed mini binders against cancer targets in just weeks, something that would have taken years using traditional methods. When tested, TILs equipped with these AI-designed mini binders showed enhanced tumor killing ability. This represents a fusion of AI-driven protein design with cell therapy that could dramatically improve efficacy. The excitement in the oncology community is palpable. TIL therapy has gone from niche academic interest to a commercial reality to a rapidly expanding field in just a few years. And here's the thing, we're still in the early innings. The field is accelerating, and the next few years are going to bring developments we can barely imagine today. So here we are. We've journeyed through the science, the technology, the clinical results, the manufacturing challenges, and the future possibilities of TIL therapies. Let's bring this all together. The core message is this: TIL therapy represents a fundamental shift in how we think about treating cancer. For decades, cancer treatment was about killing cells. Surgery to cut out tumors, chemotherapy to poison rapidly dividing cells, radiation to destroy tissue. These approaches work, but they're blunt instruments. They damage healthy tissue along with cancerous tissue. They have significant side effects, and they often aren't curative for advanced disease. Then came targeted therapies that zero in on specific molecular vulnerabilities in cancer cells. Better, but cancers can develop resistance. Then came checkpoint inhibitors that unleash the immune system. Revolutionary, but only for the subset of patients whose tumors are already inflamed and immunogenic. Now we have TIL therapies, and it's different. It's taken the soldiers that are already on the front lines, the lymphocytes that have infiltrated the tumor and recognized it as foreign, extracted them, reinforcing them, and sending them back in overwhelming numbers. It's biological warfare using the patient's own army, and it's working. Let me recap the key takeaways. TIL therapy is personalized medicine at its most literal. Every patient gets a unique product made from their own cells, targeting their own tumor's antigens. This isn't one-size-fits-all medicine. This is bespoke therapy. Clinical results are compelling. Nearly 1 in 3 heavily pretreated melanoma patients responding to the therapy, with response lasting for years. Almost 20% of patients are alive at the 5-year mark who would have been dead within months. Expansion into lung cancer, cervical cancer, breast cancer, and more are showing early promise. Manufacturing is the bottleneck and the opportunity. Scaling autologous cell therapy requires sophisticated infrastructure. Cloud-based manufacturing execution systems like Apprentice.io's Tempo MES are essential tools for managing complexity, ensuring chain of identity, maintaining quality, and enabling scale. Next-generation engineering is enhancing efficacy. CRISPR modifications, membrane-bound cytokines, enhanced trafficking, all of these are pushing the boundaries of what TILs can do. And AI is accelerating everything from patient selection to manufacturing optimization to novel TIL design. Artificial intelligence is turbocharging the field and compressing timelines that used to take years into months. The future is bright and coming fast. More approvals, more cancer types, earlier treatment lines, reduced toxicity, lower cost, broader accessibility, all on the near-term horizon. Now let's talk about the social, economic, and moral dimensions of this, because this isn't just a scientific story, it's a human story. Socially, TIL therapy represents hopes for patients and families who've been told there are no more options. The phrase, "nothing more we can do," is devastating. TIL therapy, even though it doesn't help everyone, provides another chance. It provides hope, and hope is not a small thing when you're facing advanced cancers. There's also a psychological element to receiving a one-time treatment that uses your own cells. Patients describe feeling empowered that their own body is fighting the cancer for them. There's less of the feeling of being dependent on external drugs indefinitely. Economically, TIL therapy is expensive, $515,000 per treatment currently. That's a significant sum, and it raises questions about healthcare costs and accessibility. But let's put that into context. Many cancer therapies cost $10,000 to $15,000 per month and are administered continuously until progression. If a patient is on such therapy for 2 to 3 years, the total cost is similar to or exceeds TIL therapies. And if TIL therapy achieves a durable complete response, essentially a cure, the long-term cost effectiveness is actually more favorable. So what's the message of hope here? It's this: we're living through a genuine revolution in cancer treatment. Not hype, not incremental progress, a revolution. The convergence of immunology, cell engineering, digital manufacturing, and advanced artificial intelligence is creating possibilities that seem like science fiction just a generation ago. Cancer is still a formidable foe. We haven't beaten it yet, but we're learning to harness the most sophisticated defense system ever evolved, the human immune system, and turn it into an offensive weapon against malignant cells. Every patient treated teaches us something new. Every trial completed refines our approach. Every technological advancement makes the next generation of therapy more effective, more accessible, and safer. The future where many cancers are chronic, manageable diseases or even curable conditions isn't a distant dream. It's coming into focus. And here's my call to action. If you're a patient or a family member, don't give up. If standard treatments have stopped working, ask your oncologist about clinical trials. Ask about TIL therapies. Ask about experimental options. Resources like clinicaltrials.gov and cancer center websites can help you find new trials. Organizations like Melanoma Research Alliance provide information and support. If you're a clinician, stay informed about this rapidly evolving field. TIL therapy is moving from academic centers to broader availability. Know where patients can access treatment or clinical trials. Consider referring appropriate patients to specialized centers. If you're an industry professional, a manufacturing specialist, quality professional, or technology provider, the infrastructure you build today literally determines how many patients can receive life-saving treatments tomorrow. Whether you're implementing cloud-based manufacturing execution systems, optimizing cell culture processes, or developing automation platforms, your work matters. The challenges are real, but they're solvable, and solving them scales hope. For manufacturing leaders specifically, if you're still relying on paper batch records and manual processes for cell and gene therapy manufacturing, it's time to modernize. Cloud-based manufacturing execution systems like Apprentice.io's Tempo Manufacturing Cloud aren't luxury add-ons. They're essential infrastructure for the therapies of the future. The investment you make in digital transformation today determines your ability to serve patients at scale tomorrow. If you're a researcher or innovator, keep pushing. The next breakthrough might come from your lab. The next engineering strategy that makes TILs work in resistant cancer types. The next AI algorithm that perfectly predicts responders. The next manufacturing innovation that cuts production time in half. These advances profoundly matter. Let me close with this thought. The human body is estimated to contain somewhere between 30 and 40 trillion cells. Among those trillions are the T cells of your immune system, maybe 200 billion of them, constantly circulating, constantly surveilling, constantly protecting you from threats. Inside every tumor that develops in your body, some of those T cells find their way in. They recognize something is wrong. They try to sound the alarm and fight, but too often they're outnumbered, exhausted, shut down. What TIL therapy does is gives those weary soldiers reinforcements. It says, we see you, we know you're fighting, now let us help. And when it works, when those billions of reinvigorated cells surge through the bloodstream and find the tumors and destroy them, it's not magic, it's biology, it's engineering, it's the culmination of decades of scientific inquiry and medical innovation. But it feels like a miracle to the patient who gets to live. The living army within us has always been there, waiting to be awakened. With TIL therapies, cloud-based manufacturing execution systems, enabling production at scale, and AI-driven insights, pushing the boundaries of what's possible. We're finally learning to unleash its full potential. The future of cancer treatment isn't just about fighting the disease. It's about empowering each patient's own body to win the war. And that future, it's now. In an industry where precision and compliance are paramount, Apprentice's Tempo platform offers a cutting-edge manufacturing execution system solution. It's cloud-based, mobile-first, operator-centric, with the most intuitive UI in the industry, making it perfect for today's dynamic pharmaceutical manufacturing environments and for the production of the world's most complex drug products. Whether you're working on groundbreaking cell and gene therapies, biologics, or small molecules, Tempo provides a fully validated MES system that's both powerful and easy to use. It streamlines your processes, ensures compliance, and adapts to your specific manufacturing needs. So get off of paper batch records and discover how Apprentice's Tempo Manufacturing Cloud can transform your operations by visiting us at Apprentice.io. Attention industry professionals, revolutionize your manufacturing process with the Apprentice Tempo Manufacturing Cloud, a manufacturing execution system built to streamline your operations, boost efficiency, and ensure compliance all in one powerful platform. Experience the future of manufacturing with our free 30-day trial. Scan the QR code on your screen now to unlock a world of innovation. Don't just keep pace, set the tempo as an industry leader with Apprentice. That's all for Apprentice.io's Industry Insights. I'm Frank Maggiore. Thanks for listening. Be sure to like and subscribe. Hit the bell icon for notifications when new episodes drop. Please comment on if you have any additional topics you would like for me to cover in this space, and we'll see you next week for more industry insights.